understand correctly, Al has taught you everything about variant calling, and now you understand VCF files and what the information in a VCF file is, the counts of alternate alleles, the counts of reference alleles, the genotype, the variants identified. So what we're going to focus on now is both before dinner and after dinner methods for trying to take what I would call a sort of a naked VCF file, which is just the positions in the genome where there is polymorphism and the genotypes of the individuals that were sequenced, and try and mark up or embellish those VCF files so we have some context, so we understand which of those variants that were identified by Freebase or GATK or whatever method you're using might have something to do with the phenotype that you're studying versus just variants that segregate in the segregate polymorphically in the population and really have nothing to do with the phenotype. So there's two main, this is either typically called variant annotation or variant prioritization. So prioritizing variants effectively depends upon annotations. So we're going to try and use some software and understand some of the fundamental concepts behind predicting the functional consequence of genetic variants. We're going to leverage the genome annotations that I talked to you about last night to do this. And then we're going to use some strategies both before dinner and after dinner to filter and prioritize genetic variants. And after dinner, what we're actually going to do is we're going to do some detective work and try and solve some genetic disorders in a mother-father proband trio using some of these methods to try and figure out what is the causal variant underlying these disease phenotypes. So you should have gotten an email. This is up on Sakai by now. So I already mentioned this, but the fundamental problem is how do we go, the VCF file, I don't know if you did a whole exome or whole genome, but the VCF file is essentially a big text file of every single position in the genome that has a genetic variant. The question is, how do we winnow down that file to the subset of variants that might have something to do with the phenotype? And you probably noticed that a basic VCF file doesn't contain that type of information. So you have a chromosome, you know the position, you know the reference and alternate allele, you know whether or not it was flagged as a potentially suspicious or dodgy variant, and you know the genotypes of the individual. So if you're studying a Mendelian disorder, you could, and you think it's an autosomal recessive inheritance pattern, you could use the genotypes, if you had one for mom, dad, and the proband, to just look for variants that meet a recessive inheritance pattern and filter things down that way. But what other pieces of information would you need to actually filter that down even further if you're studying a rare disease like an unsolved Mendelian disorder? You not only need the genotypes, but what else would you need? So if you think about the Punnett square, there are going to be many variants that are inherited in a recessive pattern, where mom and dad are heterozygous and the proband is homozygous for presumably the alternate allele. There's going to be thousands of those. So what else would you need? What other information would you need in this VCF file to winnow it down to maybe a handful of candidates?
Common variants. The common variants.
So and what would you do with common variants?
I know when SNP is very common in the population, but it's probably not disease-causing.
Exactly. So that would be, its population frequency would be a piece of information that you could use to filter out the vast majority of variants. So that's one thing. What else?
Change in amino acid.
Yeah, or more broadly, does it affect the gene? Does it overlap a gene at all? Right. So that's great, because that's actually, those are the steps that we're going to go through to add that type of information to answer those types of questions. So this is just some slides from the 1,000 Genomes project that describes some more details about the VCF format and some other tools such as VCF tools, which we're not going to talk about, but I wanted to include this slide so that you can go back to it later and maybe use VCF tools and other methods for filtering VCF files. So this is another example of a VCF file, and the way that we're going to actually add this information, these annotations, is in the info field. So you probably looked at the info field. Here's a basic example. You know, for the most part, what comes out of the variant detection algorithms is a sort of summary information about the types of the data that was observed at this particular locus in the genome. You know, it'll give you things like, is there evidence for strand bias? You know, were the alternate alleles observed always on the forward allele? If so, that's potentially a sign of a false positive. Or what was the total depth of sequencing across all the individuals that were sequenced? But the info field is also used, it's essentially a free form column in the VCF file where you can store really any information that you want. And that's where the tools that we're going to use actually put the gene annotations or population frequency or what have you in the info field. So that's essentially where all that information is stored. So I showed this slide last night, but the challenge is to go from an otherwise anonymous polymorphic site, bring in all these annotations, collect the information from all these resources based on this position in the genome, and put all those annotations into the VCF info field. And then downstream tools will allow you to look at the values that are in that info field and filter based on population frequency or whether it makes an amino acid change, etc. Okay, so it sounds really simple, but variant annotation actually proves to be much harder than it would seem to be at first thought. One of the fundamental problems, I think, especially in terms of interpreting coding variation, is the impact of a given nucleotide change in a position in the genome depends upon the transcript for the gene that you're looking at. So if it lies if the position of the genome overlaps an exon that is skipped in some isoforms, well then any change in that exon doesn't really affect those isoforms, but it does potentially affect the isoforms that include the exon. So really what we're dependent upon to make these predictions is, A, we have to choose a set of transcripts that we want to use to do this type of annotation. There's Ensembl transcripts, there's UCSC transcripts, there's RefSeq transcripts. So that's one dimension of the complexity. The other dimension is that we depend upon software tools to look at the position in the genome, the set of transcripts that they're using, and make a prediction as to what the change are. And by and large, it's straightforward. I mean, you're effectively looking up in the codon table to figure out, okay, this change to this change, what is the amino acid change that would be predicted? But there's corner cases, and we'll talk about that. And then, obviously, it's very, very difficult to interpret non-coding variation because we know very little about what non-coding variation does. You can do simple things like, well, is it in the first 1,000 or 2,000 base pairs upstream of a gene? If so, maybe that's the promoter. Maybe that disrupts transcription factor binding. So that's why projects such as ENCODE, Roadmap Epigenetics, Epigenomics rather, there's many, many projects that are trying to get at better predictions of the functional consequence of non-coding variation. Okay, so this paper from Davis McCarthy, I think last year or the year before, probably 2 years ago now, basically summarizes something that people that were, if you were working in this area of trying to annotate the functional consequence of genetic variants in human genome studies or in any genome, we all knew that this was a problem, but this paper does a really nice job of actually quantifying the problem. How often do different tools disagree in their prediction? How often do the predictions disagree based on different transcript sets? So I'll just point out this one highlight that it turns out to be, this is slightly pessimistic, but they found that only 44% agreement in annotations for loss of function variants when using the RefSeq and Ensembl transcript sets using the same software. So essentially, the experiment they did was they used this one piece of software, which I'm not going to talk about today, with 2 different transcript sets, and ask, based on those 2 different transcript sets from this software, how often are the predictions of a loss of function variant the same? And the answer is only 44% of the time, which is pretty discouraging. Yeah?
Can you give us explanation for this? What is the shocking results?
Yes. So the biggest difference is that RefSeq is a much more conservative set of transcripts than produced by Ensembl. So when you get into so that's sort of the punchline of the paper. When you get into the details, most of that discrepancy is for transcripts from Ensembl that were computationally predicted or have very, very little evidence of expression in different tissue types. So, you know, the punchline is that you really need to, you can use a tool like Anavar or VEP or SNPFF, which we're going to talk about in a minute, but you really have to look carefully at the transcript. You know, once you identify a set of loss of function variants in a genome that you're sequencing, you have to then do the follow-up to figure out, okay, is this transcript actually expressed? Is it ubiquitously expressed? Is it from a pseudogene? Is it, you know, what is it? And so that's, you know, if you read the paper, that's what they get into, sort of the details of that discrepancy. But Ensembl this is a much larger superset of transcript predictions than from RefSeq, and that's the big reason for the difference. But yeah, it's fairly depressing. So here's an example from this paper that I think kind of highlights the problem. So basically, we're focusing in on this gene called ABCF1. And the nucleotide, the polymorphism is an insertion of a single nucleotide, a single A at this site. And the issue is that, okay, given that we know that there's an insertion of a single A, what is the consequence on that transcript? Well, there are multiple potential answers. One is that this is this causes a stop codon, right? Because you basically, here's TG, you've inserted an A. But that's not quite true because it was already a stop codon, right? Because there was already an A there. So another option is that it could be a stop loss. Some tools might predict it to be a stop loss because you basically disrupted an existing stop codon, but that's fallacious because you've disrupted it with just another A, so you just kept the stop codon. Or you could predict it to be a synonymous change because it was an insertion of the same nucleotide, so basically no change at all. And the problem is that 3 different tools give 3 different answers. So there's no real easy solution here. So this is basically to help you understand what the problem is. Anytime you identify a potential loss of function variant, so if you ran Anavar and you found this to be Anavar predicted to be a frameshift, you really need to dig into every one of those predictions, look at the transcript, look at the actual variant, and assess whether or not that's really the consequence. Now I should say that subsequent to this paper, this kind of raised a lot of eyebrows, especially with the developers of these tools, and they've added more sophisticated logic to sort of more routinely and standardize the predictions for corner cases like this, but nonetheless, the point is that you still have to dig in and really scrutinize some of these predictions. Okay. So there's a blog called Golden Helix. There's a company called Golden Helix, and they maintain a blog that basically, one of the recent posts in the last year or so was quantifying the differences between these 3 different prediction tools. And on the y-axis is each of the different sequence ontology terms, we'll talk about that in a minute, that classify the impact of a given genetic variant. And what you see is that for each class, there's often sometimes subtle differences between the frequency with which that functional class is predicted, but there's some where there's a huge difference. So intronic variance, ANOVAR seemingly tends to say that things are not intronic when the other tools say that they actually are. Some of this may depend upon transcripts, but in this case, they controlled for that. Every one of the tools was given the same transcript set. So essentially what you're seeing here is differences in the code. Differences in the code that decide what the prediction should be based upon the scenario that's presented. Yeah?
In this example, is the total number of variants that was fed to these three programs the same? It should be.
Yes, yes.
Wow.
Yep. That doesn't quite add up though by eye because there's a big drop. It's a big drop here in intronic, but the red bars are only higher here. So we could look at the blog post to see how they handle that, but it might be that there's subsets of predictions that are not placed into this histogram. Okay, and specifically focusing on splicing variants, which are interesting because that's when you're screening for Mendelian disorders, you're typically looking for loss of function variants. So splice variants, variants that introduce a stop codon, or frameshifts that disrupt the reading frame. And so the overlap among these, in absolute numbers, the overlap in splice variant predictions from the three tools is pretty good. But there's a ton of predictions that are private to this one tool, SNP-FF. They don't dig into any further to say whether or not what fraction of that 552 are actually correct. It's just trying to convey the differences between these tools, the predictions. So I mentioned this already, but the problem is that for a given gene, especially one that we understand the function of quite well, TP53, if you compare the transcript, the isoforms presented by RefSeq and by Ensembl, there's a fair amount of overlap for this, what's presumably like the canonical isoform, but there's a huge difference in the set of transcripts between these two transcript sets. So to address this, and this is a problem that has kind of plagued not only human genetics but mouse genetics and any model organism genetics. There's a website called APRIS. The paper for this came out, I guess, a couple years ago. That essentially it tries to define a principal or canonical isoform for every gene based upon transcripts from Ensembl and RefSeq and all these different transcript annotations to define, what is the canonical isoform for each gene, so that we can, if you want to do comparative analyses across tools or across studies, you can standardize on a single transcript. Obviously, that only works some of the time, because if the transcript that is related to the disease phenotype that you're studying is not the canonical transcript, then what do you do? But if you're trying to do a comparative analysis, Jim in my lab has been using this resource to just define canonical transcripts across the human genome. It's a nice resource because you can basically take all of the Ensembl transcripts and they'll mark which ones are the canonical transcripts for each gene. Okay. So this is now built into Ensembl. So we looked at Biomart last night. You can also use Biomart to pull down or you create a data set of all protein coding transcripts, and it'll mark which transcripts are the principal isoform. That's what the PI stands for.
What does it do when, you can imagine there are times in which,
there's ambiguity.
Right, like say there's a frame shift in one exon.
Yep.
And so you really can't include all of the exons. That transcript doesn't exist,
Right. So APRIS doesn't address that. That would be a prediction made based on one of the annotation tools, modulo whatever set of transcripts that you tell it to assess a variant on. Does that make sense? So you would not, So I guess I probably didn't explain it well enough, but principal isoforms are really just used for, if you wanted to compare the predictions of two tools, you could just choose the principal isoform to feed into both of those tools and then compare the predictions among those, rather than, because some transcript sets have 50 isoforms, some only have 10 for the same gene.
It's not just a union of all exons.
That's right.
Okay.
That's right. Okay. So if you restrict, so what do you think the union would be, or the intersection of these variant impacts predicted by these three tools if we did restrict it to principal isoforms? I mean, the idea is that, the discrepancy here was pretty extreme, but presumably a lot of this has to do with the fact that they are choosing the most deleterious impact for a given transcript for a given gene. But if you just restrict the input to these tools to be the principal isoform for every gene, then do you think the overlap would be better or worse?
But that would change
Yeah.
Like the example you showed with the different, them calling the same mutation different things, that would change that, would it?
No, it wouldn't in that case. That's right. Those corner cases would still exist.
Where
Yeah.
thought those mutations took place because of whatever transcript.
That's right. So it would improve the distribution. That's exactly right. But it wouldn't solve every corner case. So that gets to another question of what does loss of function actually mean? How do we computationally predict loss of function? And there's a nice paper from Dan McCarthy, gosh, almost 6 years ago now, that sort of got at the fundamental problem here. So we tend to think of loss of function variants in 4, possibly 5 categories. So one is when there's the introduction, a nucleotide change introduces a stop codon. So that's a nonsense or a stop gain mutation. A frameshift, so if an insertion or deletion is not a multiple of 3, then you potentially disrupt the reading frame of the transcript. So one base pair deletion, in this case, would disrupt the reading frame from this point forward, and therefore probably screw up the protein product. If you disrupt a canonical donor acceptor site, then it's very likely that you're going to dysregulate the splicing of this transcript. And if you have an entire exon or gene deletion, obviously that's probably a loss of function for that protein, because you've completely screwed up the splicing process. And if the key transcript for the phenotype that you're studying depends upon this exon, then it's probably going to have an effect. But the problem is this inherent signal to noise issue. So if you think about sequencing technologies have a basal error rate. Let's just say this is 1% error. Obviously, sequencing technologies have gotten a lot better, so the error rate's lower. But we have an error rate, and then across the genome there are the nucleotide diversity rate, or the polymorphism rate across the human genome, is not uniform. So non-coding regions have more genetic variation than coding regions. So this is depicting an exon, and this is intronic sequence. So the rate of variation in the intronic regions is much higher than in exonic regions. So the point that Daniel and Chris Tyler Smith were trying to make in this paper is that in coding regions, because there's purifying selection, genetic variation is not tolerated as much in coding regions, the error rate, or the noise, is very close to the signal. So the take-home being that when you look at potentially interesting polymorphisms in coding regions, more often than not, they're wrong. Or the adage being, if you think it's interesting, if it looks interesting, it's probably wrong. Whereas in non-coding regions, more often than not, what you see is actually real, because the the signal is much higher than the noise. There's also other caveats. One is that I think the most important is that if you think about frame shifts, a frame shift disruption, at the beginning of a transcript is going to have a much bigger consequence on the final protein product than a frame shift in the terminal, 5 or 10% of the transcript. And so what they did in this is the key panel from this figure is that if you look at sort of the raw indels in blue, so this is like all the indel predictions that were found, versus the high confidence predictions, what you see is that in the terminal 5% of the transcript, you have this big boost in the raw indels versus there are very few real indels or frame shift indels in the terminal 5%. So basically, you're going to use a tool like SnippyFF or VEP that we're going to talk about more tonight and later this afternoon. But you really need to take an extra step and say, well, where in the transcript does this frame shift actually occur? And if it's in the terminal part of the transcript, then potentially it actually doesn't. It's not. It may not be loss of function. Okay. So to get at this problem, there's a nice plug-in to. If you're working on this, I encourage you to use this piece of software. It's called Lofty. And it's a plug-in to another piece of software called VEP, or Variant Effect Predictor. That's from Ensembl. And basically what this tool does, it's a script that looks at all the putative loss of function variants that come out of VEP based on the transcripts that you give it and the variant predictions that you give it. And it attempts to account for a bunch of the corner cases to essentially remove false positive loss of function predictions and basically tag predictions as high confidence loss of function variants or low confidence. So some of the things that it tries to get rid of are, for instance, variants that are in the terminal X%, you can set that, the default is 5% of the transcript, are most likely not loss of function. The other thing that it does is, did Al talk about multinucleotide polymorphisms? No? Okay. So the idea is that say you have an exon and you have a nucleotide change, let's say the reference genome is T and A, and you have a change here and a change here. The problem is that each of these changes are going to be a single record in the VCF file. What these tools that we're going to talk about do is they inspect they predict the consequence of each of these changes on their own. But what you really want to be able to do is, you can tell from the sequencing data that these two polymorphisms are on the same haplotype or on the same allele. So what this program does is goes back and recognizes that these are so close to one another that you can figure out whether they're on the same allele and adjust the prediction. Sometimes you can say, well, actually, these two changes compensate for one another. Did you have a question?
So in this example, are those right next to each other or just near each other?
Right next to each other.
So I thought that when we were going through stuff with Al, he said that when you're using freebase, if it occurred, if it always occurred together, then it would kind of pull that out together instead of one snip and another snip.
So not all tools do that.
Okay.
Yeah, so that's behavior that Freebase has, but not all tools do that. But Freebase, if you give a multinucleotide polymorphism that would come out as a single record from Freebase, it's not always the case that VEP will account for the fact, it still may look at each of those changes individually.
So even if you have it in that format that Freebase gives it to you, would you want to convert it into something that has it as two different single nucleotide polymorphisms
No,
See that?
this program will handle both cases.
Okay.
You don't have to break it up into 2 lines. It'll just recognize the fact that there were 2 nucleotide changes in the same variant prediction.
Okay.
Okay. So, how many of you are familiar with polyphen and SIFT and these? Okay, so a couple of you. So, one of the areas of research that's been, I mean, people have been working on this for almost 15 years, I guess, is trying to predict, well, actually more like 30 years, predict the functional consequence of missense or non-synonymous changes. And there's multiple tools to do this. The most widely used are SIFT and polyphen. But basically the challenge is, you know there's an amino acid change, but how likely is that amino acid change to really disrupt the function of the protein? And so there's a paper that came out, I guess in 2014, from Jay Shenduri and Greg Cooper's group that described a method called CAD. And what they're trying to show, CAD is this black line, the C score or CAD score. Has anyone, if you're not familiar with ROC curves, this is responder operator characteristic curves. Basically what it's showing is the trade-off between the specificity and the sensitivity of the method. So an ideal, a method that always predicts the right answer without ever missing any predictions would have an area under the curve that is equal to 1. So basically the line would go straight up to here and then straight over. So basically the more that the line approaches this upper left-hand corner, the more accurate the prediction method is. So what this paper is showing is that this CAD score does a better job of predicting the functional consequence of missense changes, so amino acid changes, than GERP or FASTCons or PhyloP. And then in this other plot, they do the same comparison to SIFT and polyphen and traditional Grantham scores. But the point that I want to make is that none of these tools do a great job. The area under a curve is, well, CAD scores do a fairly good job. So 90% of the time they get the prediction right. But it's still an unsolved problem. So basically, you know, when you sequence a genome, you're going to find hundreds and hundreds of missense or amino acid changes in a single individual. The question is, you know, which of those actually disrupt the protein function? And you depend upon tools like this to try and to rank order those amino acid changes. But the point here is that it's not a solved problem. We can also use conservation. This is a traditional approach. The intuition being that if there is an amino acid change, it's more likely to have a consequence on the protein function if it lies in a highly conserved exon or in a highly conserved residue. The problem with that, and that's been dogma. I mean, that's a standard method. And it works most of the time, except there's a few papers published in the last year, most recently from Jordan et al. Earlier this year, that shows that sort of surprisingly, 3 to 12% of variants that are known to cause human disease are actually fixed. That allele is fixed in other species. So that sort of debunks the whole conservation argument to some degree. So there are always exceptions to the rule. Okay, so we've talked about the challenges in sort of the methodological challenges in identifying or predicting what the consequence of a variant is. But there's also a challenge in just describing it. You know, what is the change? And it sounds a bit boring, and in reality, it is a bit boring. But there was an effort led by a colleague of mine at University of Utah and by Ensembl called the Sequence Ontology Project. And the idea is that they wanted to come up with a standard nomenclature or ontology for describing potential changes, the impact of a single nucleotide polymorphism or an indel or a structural variant on protein coding transcripts. And so this column reflects the nomenclature that they use. For instance, a synonymous variant is the label or the description should be synonymous _ variant. Non-synonymous is up here somewhere, I can't see it. Stop gain, stop loss. And the reason that this was an important project is that previously tools had their own nomenclature. So, VEP would call a non-synonymous variant maybe a missense change. Another tool would call it non-synonymous. Another tool would maybe call it something else. So when you're actually trying to compare and contrast these different methods, it becomes pretty ugly because there's not always a one-to-one relationship between the description from one tool and another. So essentially the sequence ontology project solves that problem by providing a standard nomenclature that now, thankfully, all of these tools use. And it's based on an ontology, so a stop gain or a stop loss prediction is a subset of a non-synonymous variant, is a subset of an in-frame variant all the way up the chain. So there's just sort of a hierarchy in the way these terms are used. So someone earlier, I think it was you, mentioned that we really need to think about population frequency as well when we interpret a genetic variant. So, the clearinghouse for all known polymorphisms, not just in human, but in other species, is called the Database of Single Nucleotide Polymorphisms. It's a bit of a misnomer at this point because it includes other types of polymorphism. But from a really simple perspective, if you were studying rare diseases, you could just ask the question, well, is the variant that I see in this family or this set of families in dbSNP? And if it is, it's not interesting to me. So that was actually done, that process, that filter was done in the first couple of studies that looked at using exome sequencing to study Mendelian disorders. The problem with that now is that dbSNP contains really rare variants, so 1,000 genomes and sequencing of lots of exomes and genomes. There are variants that are in dbSNP but are very, very rare. So if the disease that you're studying has a prevalence of 1 in 1,000, you don't necessarily want to throw away any variant that's in dbSNP, especially if it's in dbSNP but only at a frequency of 1 in 10,000. That's actually a potentially interesting variant. So there's kind of been a movement in human genetics, especially for rare disease genetics, to not really use dbSNP for that purpose. Just use dbSNP for informational purposes, but filter or prioritize variants based on their allele frequency, not just their presence or absence in dbSNP. The other problem with dbSNP is that it actually contains somatic mutations from cancers, which obviously you don't want to filter out in studies of heritable disease. So one of the first data sets that sort of trumped the use of dbSNP for rare disease genetics was the Exome Sequencing project. So this was a project funded by the NHLBI to sequence 6,500 exomes, meaning, so essentially we've got 13,000 chromosomes for nearly every protein coding exon for every protein coding gene. And what they found is that nearly 45% of the variants that they discovered among these 13,000 chromosomes were present as a heterozygote in one individual. So their frequency was 1 over 13,000. So there's just a huge excess of really rare genetic variation in the population, primarily owing to massive population growth over the last 100,000 years. Well, this didn't really show up, but the Exome Sequencing project has a really nice resource called the evs.gs.washington.edu, and it's basically a clearinghouse for every single genetic variant that they observe for every protein coding gene, its frequency. They color code things based upon whether it's non-synonymous or synonymous. So you can use this to basically ask, was my variant seen in the exome sequencing project? More recently, we've probably mentioned a couple times a larger study called the Exome Aggregation Consortium. And it basically, it's the exome sequencing project, but on steroids. Now, we're talking about 65,000, not 6,500 exomes. This is work led by Daniel McCarthy, and I think actually in-house at the Broad Institute, they're analyzing over 100,000 exomes. But what's released to the public is a subset of 65,000 exomes. And what's really interesting to me is that the Exome Sequencing Project found that 45% of the variants that they observed were present on 1 out of 13,000 chromosomes. In this study, it's now over 60% of the variants are a so-called singleton. It's a single heterozygote among 65,000 individuals, or have a frequency of 1 over 130,000. So it's just this huge excess. And if you read the paper, one of the main claims in the abstract is that this represents out of every 8 base pairs of protein coding sequence, there's a genetic variant 1 out of 8 times across 130,000 individuals. And they have a follow-up paper that I haven't read yet that predicts the density of genetic variation that will be observed when we get out to 500,000 or even 1 million exomes.
So, can I
Yeah.
on basis of these two sort of data points, 6 out of 565,
Yeah.
can you estimate how many people you would need to exome sequence before every variant was?
So that's what this paper that they just submitted gets at, is sort of when do we reach the saturation point? I don't know what the answer is, but yeah, I think that's what they attempt to do in that paper actually. It's on bioRxiv. So yeah, I mean that's kind of a critical question because you sort of want to know when to stop. So the EXAC consortium sort of learned from the exome sequencing project, and that really to get these types of resources widely used by the community, as they should be, you need to make really convenient, nice interfaces to get at the data. So if you go to EXAC, I think the URL is the same, exac.broadinstitute.org, and type in your favorite gene, what you'll get is this is a fairly busy readout because ATM, one of my favorite genes, is a really huge gene, but basically what you see is all the genetic variants that were observed in that gene across 65,000 exomes. They continually update and improve this website and the resource, but you get this HTML table which gives you every single variant, where it's, whether it's in a 5' UTR or in an exon, or if it was non-synonymous, etc. And then it gives you an estimate of its allele frequency among those 65,000 individuals. So obviously a data set like this is crucial for prioritizing variants in studies of rare disease and common disease, because you basically can look at the allele frequency and make some guess as to whether or not it has any relevance to the phenotype you're studying based on the rarity of that phenotype. It also gives you other metrics such as the population, the frequency in different world ancestries. So the overall allele frequency in this case is 2%. So 2% of 130,000 chromosomes, so that's what, 2,600 chromosomes had this allele. But you notice that there's population stratification of that frequency. So some world populations, so in Europeans it's even more prevalent than 2%, but in East Asians it's a quite rare allele. So that's important if you're studying Mendelian disorders in an East Asian population and not people of European ancestry. Your interpretation of this allele and how you filter it would change pretty dramatically. That's what, 2 or 3 orders of magnitude difference in frequency. So that brings up another point, is that you really can't filter genetic variants based on their overall allele frequency, or you shouldn't, especially in the study of rare disease. And the point that I want to mention is that if you did that, this is a case where this allele T at this position is present overall across all these world subpopulations on 4 out of 1,000 chromosomes. But the intuition behind my argument is that if you're studying a rare disease phenotype, typically they have a highly penetrant deleterious effect on proteins. So if that's the case, that variant should be rare in every single population, right? Otherwise, I mean, unless there's some very extreme modifier, for instance, at another locus in Finnish genomes, then if this variant actually has something to do with the phenotype, it really should be rare in the Finnish population and every other population. So the strategy, the correct strategy, I argue, is that you shouldn't filter on the average allele frequency across all populations. You should require that it's rare in every single population that you have access to. Okay, so how do we go about annotating variants from a computational perspective? There's a piece of software that I developed as a postdoc and that we maintain in my lab called Bed Tools. We'll talk about this tomorrow. But the basic idea is this gets back to the whole interval concept, genome interval concept that we talked about last night. So imagine that we have a set of polymorphisms in blue and genes in red. Essentially, the process that all these tools use is we first ask, give me all the variants that overlap or intersect genes. And then once we have that collection, we go through and try and predict what the consequence might be. But Bed Tools and other methods, they use genome intervals in any format. So we talked about Bed and GFF and VCF and BAM, et cetera. So you can use this class of methods not only to look at what variants overlap genes, but you could use it to count how many sequencing reads were observed for that individual or at that site across your study. And there's all sorts of other methods that you can apply, and we'll talk about those tomorrow. So for instance, overlapping genes would be this simple command where you take your VCF file and you tell it to intersect your VCF file against a bed file of genes. You can also you can quickly count how many intersections there are. And we'll go into much more detail on this tomorrow. But basically, all of these tools effectively are using what I call genome arithmetic. You're taking these genome intervals in various formats and slicing and dicing and comparing and contrasting them to collect annotations. So the tool that we're actually going to use and spend the last little bit working on is a method called SnippyFF. The reason we're using it is that it's very, very simple to use and it's very fast. There are other more sophisticated methods that we're going to talk about after dinner. First and foremost, a tool called VEP. I've mentioned it a couple times. The problem with VEP is that it's pretty hard to install and takes a long time to run. So it just doesn't really suit itself well to what we're doing interactively. But we're going to use SnippyFF as a demonstration of what these tools do. So we're gonna go a bit wild, and we're going to try and actually install this software ourselves on each of our Amazon machines and run and annotate a VCF on our own so you guys can actually see how this tool works. Okay, before we do that, I just want to talk about what it does. The main feature, the other main reason to use SnippyFF, especially if you're not studying human, Pablo Cingolani, the guy who develops this, has done a ton of work to collect all the annotations to be able to do these types of predictions essentially for any species whose reference genome has been sequenced. So it'll work for any model organism or species that you're working on. VEP is a little more tuned. It's not exclusive to human, but it's really most well suited to human. Okay. So basically what SnippyFF does, as we talked about, is it starts with a naked or raw VCF file from Freebase or GATK, and the output is in this info field. So in this case, what it's saying is that this variant and this variant are both predicted to be a stop gain. That is predicted to be a high impact. And here's the codon change and the amino acid change. And it provides more information than that. Effectively, it'll try and make a prediction for every single transcript. This is just an example from their documentation. So the output of SNP-EFF follows a formula. They first define what the effect is, the impact. They give you the codon change, the amino acid change. This is just really here for your reference. But these big hideous strings that you're going to see that get put into the VCF file, they actually follow a structure that you can look through by eye, or downstream tools will know that the 1, 2, 3, 4, 5, the fifth field is the amino acid change, for instance. Okay, so now what we're going to do, I tested this out a few times beforehand and it worked fine, so hopefully it'll go smoothly. We're going to download and install SNP-EFF, and I lied to you, we're not going to do it on Amazon, we're just going to do it on our local machine. The reason for that is there's not a convenient way to download SNP-EFF up onto Amazon. We're just going to run it locally. It's just easier. We've run into more problems. So if you go to the website, which is right here, you should be able to just click on the link from the PDF. The first thing you should see is this download button. And when you click download, it should bring you to a SourceForge page that will sit, and it'll look like it's doing nothing, but after 10 or 20 seconds, you should get a dialog that looks like this, where you can actually save a file. So I'm going to give you, just wait a minute and just see if anyone has trouble getting to this page, to this dialog. Bless you. Has anyone seen something like this yet? No? Let me take a look around and see what's happening.
So mine downloaded.
Yeah, they downloaded.
Oh, it downloaded? Okay. Where did it go? Downloads folder? Okay. Okay, so I actually would rather we do things on the desktop if possible. So why don't you either use a Unix command to move the file that was downloaded to your desktop, or you can use Finder to drag and drop it. So essentially the file is, you're looking for a file called SNP-EFF _latest _core.zip.
So our files aren't zipped, we just let them through there.
Oh, it just opened it up? Okay.
Yeah, so yours was just. Yeah, yeah.
Okay. So I guess some people's browsers automatically unpack the zip when you download it. Okay, so if that happened, and I think that'll probably happen reproducibly for everybody, what you want to do is move that SNP-EFF folder to your desktop. Work for you? Good? Yeah. Okay, once you've moved the SNP-EFF folder to your desktop, you can essentially skip this step because for most of you it's already unzipped. But you basically just want to go into that directory. So go to your desktop and then change directory into that SNP-EFF directory. And once you're in there, if you ls to list the contents of that directory, you should see a bunch of files, primarily one called snipeff.jar.
So for people who don't know how to get to the desktop, if you just go CD, you can drag the folder right into terminal, and that'll automatically put in the right.
Cool. That works.
Yeah, and you can't do it in your Amazon cloud. The term may never open with Amazon cloud.
Yeah, you need to be doing this not on Amazon Cloud, but on your local computer.
No, so start a new window, so go to shell, new window.
So if anyone's having trouble, just raise your hand because we've got plenty of people to help out. I'm happy to help. Can you help out if anyone's having trouble? Oh, okay, yeah. No, not yet. Don't move on to these steps until we get everyone with SnippyFF in their desktop directory. Does anyone need help? I'm happy to help. Good? Okay, so we're not gonna run these commands yet, but I just want to explain first what's going to happen. So this snippyff.jar file that should be in this snippyff directory that you have on your desktop now, that's actually the program that actually does all the annotation. So the .jar, this is a convention for programs that are written in Java. They tend to have a .jar extension. But the first thing that we need to do for SnippyFF to be able to annotate a VCF file, it has to know what species you're dealing with and what transcript set you want to use. Not only that, it has to know a really crucial piece of information is what version of the genome you're actually using. So the VCF file that we're going to use came from sequence alignments that were aligned to build 37 of the human genome. So that's the coordinate system. So if you think about what these tools have to do, it's A, they have to know where is the variant on the genome, and then B, they figure out, okay, based on that position in the genome, what transcripts overlap that position. So the coordinate system really matters. So things would be really bad if we called our genetic variants using Freebase or GATK using build 37 of the human genome, and then we used annotation, we told SnippyFF to use annotations based on the build 38 coordinate system, you'd get some pretty wrong answers. Basically, every prediction would be wrong. Okay, so what we're going to do is we're going to ask SnippyFF to download build annotations based on the Genome Reference Consortium Human Genome build 37. So the 37 is for build 37. And 75 is the Ensembl transcript version. So Ensembl is, I think, up to build 82 or 83, but we're going to just use, yeah.
I think some of these don't have Java.
Oh, really?
Yeah, I was just trying to do it on the.
Yeah, that's a bummer. Well, that's okay. If it doesn't, well, let's see if anyone has Java on there.
Anyone getting it? Or no? Yeah.
No Java? Okay. Well, no, I think what we'll just do is I'll walk through it and I can show you the examples. It's just, we were just trying to see what the annotations look like. So I'll walk through it. Oops. That's too bad that we don't have Java. Okay. Okay, so this will be a follow me exercise, or watch me rather. Okay, so maybe I can make this a little bigger. Okay. So this command is going to download all the annotations that it needs based on Ensembl build 75 for build 37 of the human genome. And this should take a minute or so. Then what we're going to do, we're going to download a VCF file that I have up on a lab a lab storage for basically chromosome 22 for, this is from a trio, mom, dad, and affected proband. And I just pulled out the variants from chromosome 22 just to make things fairly small and simple. Okay, so while that's running, I'll show you what that file looks like. So obviously it's a VCF file, so we have all the VCF header information, which I know you've gone through. And then once you get past the header, which is huge, okay, now we start to see individual polymorphic sites on chromosome 22, not surprisingly. But the point is that the info field is either empty or very sparsely populated for each of these records. We're going to run SNPEFF to actually populate all the annotation based on transcripts that we want to be able to filter and prioritize these variants. Okay. Oops. Okay. So what happens once it's done downloading, there's a data directory. And in that directory, you see grch37.75, which is the data set that we asked it to download. And what you see in there is just tons and tons of files that SNPEFF uses to do all this annotation work. So if you're working on armadillo or a mouse or whatever, you could basically figure out, if you go to the SNPEFF website, it'll tell you the name of the data set that you want to download. So I don't know what it would be called for armadillo, but maybe looked like there was one build of armadillo, so maybe it's ARM1 dot something for the transcripts that are there. But the nice thing about this tool is that you can do it for any species for the most part. You should check to see if it's actually there. I don't know if it is. Okay. So now we've downloaded all the annotations. I had already downloaded this VCF file, and our goal is to run SNPEFF. So what we're going to do is run SNPEFF using that annotation database against this naked VCF file and produce a file that has been fully annotated with all the transcript, based on the transcripts that we asked for. Yeah?
What is the XMX?
Oh, yeah. Good question. So Java programs, this program has to be given a certain amount of memory. And basically this string is telling it effectively how much memory to give that program. So it's saying, I want to run this program using Java, unfortunately, which you don't have on your computers, but I do. And you want to give this program 4 gigabytes of RAM to do its work.
What if you don't?
The default varies. I think the default is still 2 gigabytes of RAM. So tools such as GATK and Picard, which I think you probably talked about, they are also written in Java. And one of the most common errors that you get when running those programs is that you just didn't give them enough memory. And it'll tell you, you'll get an error, and then you go in and you change this variable to give it 4 or 8 or 16 or whatever, how much memory you have, until it stops giving you errors, essentially. Okay.
There's actually database commands, so you can see.
Oh, which is there?
Yeah.
Is it there?
No, but you have to run it.
Oh, there's a list on there. It's buried in that website somewhere. There is a list of everything. I just don't remember where it is. Okay, so now I'm going to run this command. And it tells you what it's doing. Some of the information is superfluous, but I'm reading this database for genome version 37.75. It tells you what file it's reading from. It builds, I think it's going to say something about an interval forest, but basically that is, it's building a big collection of all the genome intervals that represent all the transcripts. So we talked about genome intervals last night. It's using the same concept to find variants that overlap transcripts, genes, exons, introns, etc. And then at the end, it gives you a list of everything of what it found. It makes some web pages, so I'll quickly show you that. Where did that go? Right. So it makes this little HTML page, which doesn't show up very well here, but it's basically summary statistics of all the different annotations that the types of variants that it found. Most were SNPs, not surprisingly. There were some insertions, some deletions, no multinucleotide polymorphisms. Most of the variants were predicted to have a low impact. So these would be cases like a synonymous change. A high impact would be things like frameshift or stop gain. So obviously they're rare. We shouldn't see lots of them. So you can use these types of statistics to get a sense of whether or not your data set makes sense. If you see the complete opposite, where most of your variants are high impact, that's probably a red flag that there's something really wrong with your database, with your data set, or potentially that you're using the wrong version of the human genome annotations for if you align to 37 or using annotations from 38, that would cause it. Yeah, did you have a question?
I was wondering what's modifier.
So modifier, they have a whole website on that. That's a good question. Let's go take a look. It's not a simple answer. So there's a bunch of different sequence ontology terms for that. Okay, so documentation. See if we can find it. If I can spell.
So hypothetically, if we download the genome, how long would that take?
The human genome?
Well, 37, the one you're going to have to download. Say we install Java on the computer.
I think it took, when I did it this morning, it was like 5 minutes or so.
Okay.
It's a pretty big data set. All right, so let's see if I can find this. Modifiers. So here, it's sort of, let's see, that's one example. That's not a very good one. Let's find this. Okay, so a downstream gene variant. So what they're trying to convey by modifier is that this variant might have something to do with either the regulation or expression of this gene, but it's not directly overlapping the gene. It's downstream. They probably say the same for just something that's exonic, but they don't know what change it makes. Intergenic, it's basically, the modifier is used to, it's like punting. They don't really know. They don't know how to rank it.
So they could actually be even in this sense.
So the exon variant, yeah, we'll have to look at how that actually comes about. So we can go to sequence ontology. So I don't know why you would ever have this, because if it's an exon variant and you know the nucleotide change, you should be able to predict with more granularity of what the actual change is. So this is probably not a term that is often used. So anyway, there's all sorts of summary information that you can go through on the website or the web page that SNPBFF generates. But let's look at what the output looks like. So this is the command we ran. The input was this naked VCF, and now we have this dressed up VCF from SNPBFF. So we can take a look at what's changed. Oops. Okay. So now what you'll notice, we looked at the first two variants before, and the first variant, if I remember correctly, had nothing in the info field. And now, it's a big string of gobbledygook, but there's this ANN in a long string in the info field. And this ANN is the output. That's the annotation output from SnippyFF. And so this first variant is predicted to be a modifier for this gene. So that's the Ensembl gene ID. So let's just scroll through this. That's the Ensembl gene ID. It's a modifier for this transcript, which is a transcript of this gene. And for other transcripts, it's predicted to be an upstream gene variant. And it just goes on and on and on. And the reason that there's so much information is that it's making a prediction effectively for every transcript, for every gene that the variant is nearby. So let's try and see if we can find one that's more interesting. Let's see if we, the website said that there were 74 high impact variants. So let's just do a quick control. How could we count how many variants are high impact in the VCF file? grep. Yep. So I predict that we'll get a number that is different than 74 just because it's never that easy. Let's take a look. Okay, that we get high, that's good. 34. Weird. Wonder why it's 74. Why do they say 74? I'll try and figure that out. It could, I think what they might be counting is the total, yeah, so basically this is only returning 34 lines with the word high in it, but there are multiple transcripts with a high impact per line, right? So let's take a look at why this variant, this last variant, is predicted to be high impact. Does anyone have a guess just looking at the variant Y? So this is the position, this is the reference allele, this is the observed allele. Frameshift what? Deletion or insertion? Right. So basically this extra G allele is predicted to disrupt the reading frame of that transcript at least, and this transcript. But, let's see, for this next transcript, it's a modifier of this transcript. So I think what we should do is go look at this position in the genome. We don't want armadillo anymore. Oh, thank you. And it's 38 by default, so we gotta change it. And then what we can do is just, I think I can just look at my computer. So we have to do, it wants to read specific format. So you type chromosome, start position, and then end position. So we'll just go to that specific site, and then we'll zoom out. Okay, so let's zoom out. That's not working. Okay, so now I'm just going to change what we're looking at, the tracks. Okay, so I told you that we were using Ensembl transcripts. So what, by default, UCSC is going to show transcripts predicted from UCSC genes, but we don't want that. We want to compare apples to apples here, so let's switch, turn this off. We'll say full for Ensembl. Okay.
So when you are typically looking at variants, do you usually use Ensembl? Do you have like a favorite transcript?
Most of our work is based on Ensembl transcripts, yeah. Just because it's a more, so that our mindset is more focused on rare disease, so we're really worried about false negatives. We're worried about missing things that could be deleterious. We're perfectly happy to go in and inspect and rule out false positives, but you don't want to miss things that are potentially there. You spend a lot of time doing that, but okay, so the position was 513. So that's right here. Yep. So in this case, it's predicting that it has a frame shift on transcript that ends in 9492. So let's see if we can find that transcript. That's this fourth one down. You see it? Okay, so let's make sure that makes sense. I guess the frame shift would be right there on that transcript. But then for another transcript, where was the one that it said was UTR? So this transcript that ends in 912, it says as a modifier, and that seems to be right because if you notice, this is a little bit thinner than this thicker part for this transcript. That's a UTR. So it's basically saying it's not high impact for that transcript because it just overlaps the UTR, not the protein coding exon. Okay.
Do we really think that's an intron right there?
We could zoom out and see.
It's just so small, isn't it?
Well, I'm only looking at a 100 base pair window, so we'd have to zoom out to give some more context. Let's go out to 1,000.
Well, I mean, it's predicting like a 2 base pair intron right there.
Ah, yeah, good question. I see what you're saying. I thought you were talking about the second line up from the bottom. Yeah, I mean, that's the prediction, but it doesn't really make a lot of sense, does it? And therein sort of lies the reason why you have to inspect each of these candidates by eye. Okay, so for the next 20 to 30 minutes, what I'd like to do is tomorrow we're going to spend a lot of time on the command line and doing a lot of Unix operations. So I'd like to, let's just come up with 4 or 5 questions that we want to ask of this data set and try and figure out what Unix commands we would use and how we would actually answer the question. So now we've got a fully dressed up VCF file. We've asked a very basic question, which is how many variants are high impact. Is there any other questions that you guys that come to mind when you think about this data set? We could try and reproduce. One that would come to mind is what is the distribution of high, medium, low, and modifier impacts? Should we look at that? All right, I'll try that first. And then what I will do, I'll answer a couple questions, then I'll put this file up on a URL so you guys can download it and actually play with it. Okay, so low. So there's 1,000 records that are have low impact. What if we wanted to know what percentage of the variants were low impact? How would we answer that? We know the actual count. But if we want to know, is it 50% or is 1,000 70%, what other information do we need? Right? So how would we get the total? In the file? Yeah, we could do that. That's not quite right. All right, so there's what'd you say? Right, so there's 2,572 lines, but there's a header, which looked like about 1,000 lines to me. So let's get rid of the header. So we can do that by saying grep-v, which means only return lines that don't match. We want to exclude any line where the first character is a £. So this caret says, I want the match to be for the first character, the beginning of the line. So let's make sure that what comes out, oops, I got to tell it what. Okay. Huh? Oh, geez, sorry, I'm not paying attention. Thank you. Okay, so, right, so let's take a look. Yep, the first thing that came out was a non-header line, and it's the first record. So now let's figure out what the real denominator is. So 2445 and we're 1031. %. Okay, so let's figure out.
Can I ask just one quick?
Yeah.
I get the grep not. I get that.
Yep.
And you did something where it was like, yeah, the little caret and the hashtag.
Oh, yeah. So, okay, right. All of the header lines, one thing I know is every single header line in the VCF file starts with this £. So the reason, so you could argue that this command would work just fine if I just said grep -v, meaning exclude, quote, £, without this caret. The problem is that if any of the lines have a £, anywhere in the record, it'll also exclude those. So if, for instance, SnippyFF decided to put a £, in one of its annotations, then that would also exclude this. It would exclude those lines. So we're being a bit more strict in saying, well, not only does it have to have a £, but it has to be the first character, which defines a header line.
And that caret is the first character.
That's right, yeah. Okay. So what if we wanted to know how many variants are not SNPs? Any idea how we'd figure that out? Besides looking at the HTML website that SnippyFF provides for us. So let's take as a hint, let's just look through. Let's look at some actual records. So looking at these records, can you by eye tell which ones are SNPs and which ones are not? Okay.
Length in the second column is greater than 1.
Yep. Let's try that. I think you mean, well, the fifth column you mean, right?
What if the reference is listed as CAG and then
Okay, yeah. Okay, so, yep, it's not quite that simple, right? So so if you had a multi-nucleotide, the reference could be CG and the alt could be GC, and that would meet our criteria. So one thing we could do, let's just be a little more sophisticated, and we will say, have you learned about awk yet? Okay. All right, well, I'll explain what's going on. Awk is a really nice program for filtering records based on more sophisticated criteria than just like string matching, like in grep. So I'll explain, but first I got to cheat and remember what column numbers are. Okay, so the reference allele is the 1, 2, 3, 4th column. And the alternate allele is the 5th, right? Okay. So what we can do is say awk. What's the first rule we want to apply? Let's say that again. The length of the reference allele? Yep. Okay, let's do that. So we say $4. So for the way we say the 4th column, we're basically saying we want the 4th column. You have to give it the $sign to know that you're talking about the 4th column, not the number 4. And we want that length, if I can spell, jeez, equal to 1. So let's just test that, see what it gives. So we should not see any records with the reference allele being longer than 1. But going through by eye, that's kind of weak, right? So let's now cut out that 4th column. Oh. Okay, now what we can do, we've got this. So this is the reference allele for every single site, every position. Now we can just build up a little histogram to see how often each allele occurs. And you can do that with, you got to sort it, and then unique. I'll explain this in a second. So now this is every possible reference allele that was observed. So we see that no allele with length greater than 2 was observed. So that's a control we can do to make sure that the command actually worked. So what's happening is, just walking through this, we're filtering out the header lines. Then we're saying, well, awk restrict. We only want to look at lines or variants where the reference allele is 1 nucleotide. That's this. Then we take all that output and send it to this command, which just pulls out the 4th column, cuts it out. So it's like, you know, if you had an Excel spreadsheet, it's just like, give me the 4th column. We then sort that. So this is basically going to, since it's length 1, this is going to be A, C, G, T, or possibly N. All this output. Then we're going to sort those values so that all the A's come together, all the C's come together, all the G's, all the T's, and if we add N's, all the N's. And then this command just says, what are the unique values that we see? And it requires that the data is sorted so it can quickly figure out what the unique values are. And it gives you a count. There were 4 unique values. All the A's came first. That's why the output comes out first. And there were 492 A's in a row. There were 665 C's in a row, 724 G's, and 487 T's. Does that make sense? Okay.
Can we see what it would look like if you didn't have that off length? You know what I mean?
It's probably the same because I don't know if there are any deletions. Oh no, I think the report said there were deletions, right?
Then I'm thinking what we'll see is the same thing, ACGT, but then there's going to be a few other.
That's right. So let's make a guess. So good questions. I think it says there's 77 deletions. So we should see something that adds up to 77, hopefully. So let's do that. Yeah, so there's a bunch of long non-length 1 reference alleles. Let's see what they add up to. So let's just make it now ones that are greater than 1. Okay, so now we can cut out that first. Wait. I'll explain what I'm doing in a second. Okay, so sum of lines 77, which should match. Yep, matches the deletions. So basically all I did there was print this extra little bit here is to just print this count column. And then I send it to a little program that I wrote to compute statistics on a bunch of numbers. So it's there are 77 records that are deletions, which matches what SNPFS says, which is good. Okay, so back to the original question, which I can't remember now, which was how many SNPs there were, right? So this gets us our SNPs, but we don't know. This possibly includes insertions, right? Because we're not requiring that the alternate allele is also length 1. So now we can just do something simple and just say we know how to filter for the reference allele being 1. So what would I write to also require that the alternate allele is length 1?
Length $5.
Yep.
And the double ends mean.
Yeah. Okay. Good. So yep. It's a Boolean and. So this restriction must be met, and this restriction must be met.
Is there other ways to say or?
Yep. So or would be. This is all the challenge in programming is just learning the syntax. You know in your head what you want to write. It's just how do the languages differ. So or would be pipe. So okay. So did the numbers change? It looks like it.
They all got smaller too.
Yep, that's good, right? So what we should do now is steal that other command I used and figure out what the total is. And it should certainly match the number of SNPs in that web page. That's 2292. Yep, it matches. So that's good. That's good. So this is one way of doing it, or you just look at the web page.
Can we ask a question of how many SNPs are in the same gene? How many genes have more than one SNP in the same gene?
We can do that. That's definitely a challenge question. Let's see, how would we do that?
Can you
Yeah.
go back to what we're looking at?
Yes, we can do that, definitely. Huh?
I was just saying, can you go back to that?
Yeah, so there's a really clean way of doing this, and then there's a hacky way that'll take less time, so I think we'll choose the latter. But, so yeah, I mean, we don't, somewhere in here there should be the no, we didn't run it to ask for the HGNC gene name. So like a gene name like TP53 is not going to be in here. It's all these Ensembl gene IDs. But we can still get a count per Ensembl gene ID. Okay, how would we do that?
Well, I see, since it's more than one Ensembl gene ID, but refers to gene, it's really hard.
Yeah,
Hey, are all those in the same column?
they are. Yep. So let's. We may not be able to solve this, but let's. I may not be a good enough programmer, but I think we can work on it at least. I forget what number it is. 7. Yep. No, 8. Okay, so that the 8th column is our big crazy info field, right?
So can you unique just like E, S, and G star?
Yeah. So one thing we're going to want to do is we basically want to pull out the.
I think the HGMC gene ID is right after the modifier. That
Oh, it is?
LTD,
Oh yeah. Okay.
So above, when you were looking at it, that AP whatever was the Vega gene.
Okay.
For whatever reason, they used the Vega annotation.
Oh, I see. Okay. So the question is, effectively it's the same problem, is how do we pull out just this bit or just this bit from every record, especially when every record has gene information in it multiple times. I got to think about that for a second.
This is a consistent number. You just turn all the commas into tabs and put them around fields.
That's exactly right. That's the problem. So, but the info field, there is structure to it. Each prediction, if you notice, is separated by a comma. So A and N, then there's this big long string with all the pieces of information for that transcript.
So then couldn't you just keep that and then tell off that your separator is now commas? but it's
Yeah,
not?
so that's
But
basically,
would you not necessarily have the same amount of like variance in each?
so yeah, so here's, I think,
Assuming that every other transcript just points back to the same gene, then just take the first gene you see. Can you do that?
that might, I think, let me show you what my idea is and we'll see if we agree that it's, I think that'll work too, but let's see. Well, I gotta cheat again. I just always forget the column numbers. Okay, so basically the problem that we're gonna face is that if we break this record up by the comma and basically expand out this record to multiple lines, we need to know that a given line went to the same variant, right? So we need to also keep track of, we'll just keep it simple and just keep track of the chromosome and coordinate. Okay, so what we're going to do is cut out the chromosome, coordinate, and that record field. That worked. Okay, now what we need to do is there's not a clean way, there's not a fast way to do this with awk that I know of, but there's a program in Embed Tools that will do this. I don't remember how to run it. How do you do it? Oh yeah. Yep, okay. So this expand program will, let's take a look at what it did. So we said expand the third column, and the reason it's the third column now is we've already cut out the chromosome. We've reduced the 10 or 12 column output in VCF into 3 columns: chromosome, position, and then the info field. And then basically what we're saying to bed tool, this expand command, is it just looks for, by default it looks for a comma. So we're telling it to look for a comma in the third column, and anytime you see a comma, break up everything before that comma into a record where you replicate the first 2 columns that were given with each of those lines. So now we've got, for this variant, apparently this variant here overlapped a whole bunch of transcripts. So what we should double check, because I wrote this code, it could be wrong, that there's a different transcript for each line. And that appears to be the case, right? Okay. So now the nice thing is that we have in the, we can figure out what column in this field is the gene. So that's one, this is going to be more challenging than I thought. 1, 2, 3, 4 is the gene name. And it's separated by pipes. So now, did I miscount?
It's got a logo.
Okay. So now what we can do is replace the pipe with a tab. So there's a couple ways to do that. Have you guys learned how to do replacement? No? Okay. There's a really simple way, and that's called TR for transliterate, which, look it up. It means basically transpose one value with another value. So now we can say we want to replace a pipe with a tab. And the way you say tab is slash, or is that a backslash? I think that's a backslash T. See, and it's all blown out into tabs, right? So now what we should do, what columns do we want to cut out now? We probably still want to keep these, right? 1, 2, 3. Yeah, so let's do 1, 2, 6. Okay, so that seems to be working. Now what do we need to do? So it's a little tricky, right? So what if there are more than just this site affecting this gene? And it probably is, right? So here's the case. So how do we handle this?
You would like to keep both of those, but maybe collapse every instance in which it says when the gene name and the number are the same.
Yep.
So can you do when both things are?
Yeah, can you do unique for 2 columns, or is it always unique for only 1
Yeah,
column?
unique across a column.
Pardon me? I don't know. Go ahead.
Can you please speak?
So basically create a field, a column that's a combination of multiple things. Okay, so what you're proposing is to make a combination of this, this, and this together.
I mean, the first column would be on 22, right?
Right. So really just these 2? Yeah, we know that's true. We do know it's always chromosome 22. Okay, so let's try that. So what's that?
I mean, you can use Excel.
Could do that. Could do that.
Press on. Don't listen to him.
I mean, so here's, if you're at your lab, there's a really nice command. So you can just take this output. There's a command called pbcopy, which will put that output on your clipboard. And you're exactly right. We could just go into Excel and answer the question and just if you hit Control V, that output that you just got on the command line will be pasted into Excel. But let's not do that because I hate Excel. So the idea, what was the proposal? To basically create a meta field that's the combination of the second and third columns. Is that right? Okay, so you can do that with awk as well. you would just say print. So now we have to, anytime you're printing, you have to use a curly sign. We didn't do that before. So we want to print, and you were saying, essentially, let's do 3 and then 2. So we want the gene first and then the position, just so I think it's a little bit easier to read. But let's maybe put an underscore so that it's a little easier to read. Okay? So now we have gene name and the position. Okay, so now we can do a unique on that. That was your proposal, right? Okay. Oh, but there's a problem. I need to sort it just to make sure that it's right. Okay. So, what's going on here? Right. Okay, so now what we want to do, we want to filter those out. So let's get rid of anything that starts with an underscore. So how would we do that? This is going to be the world's longest Unix command. I think we might set a record here, actually.
The caret sign.
Yep. So grep, and then what? Yep, and then what? Right. Caret underscore. I think that, yeah, that worked. So now we've got genes, okay? But now what do we do? Now we have to figure out, now we want the, so we've got it distilled down to the distinct positions, but now I think we ultimately wanted the count per gene. So how do we get that? This is kind of fun. What's that? Yeah, so basically we now just want the unique, the count for each gene. So we got to basically break this apart. So how do we break it apart?
TR.
Right, TR. And then what do we want to replace it with? This is getting ridiculous. Okay, there we go. Now let's sort that.
When you do that TR command, it's only applying it, whereas it gives you the output and it's also changing the file, right? Where's this file?
There's no file.
All we're doing is output.
Yep, we can make it a file, now. We can just land it to a file. And now we have a file called foo that we want to save, and we can look through. But unless you tell Unix to create a file, it's just going to dump stuff to the screen, or called standard output. Yeah, what's up?
Can we just grab before after the sort and then do unique dash C?
Probably. We're just working through it. Potentially, there's a couple of easier ways to do this, but I think it's fun to just work through a solution. Okay, so now what we do, you've already done it? Oh, I thought someone had said it worked.
There's a more efficient way. The process of watching you do this is extremely helpful.
All right, so sort, and now we're going to do unique. Oh wait, no, I blew it. We got to cut out the first column, right? Well, let's just see what that looks like. Yep. Okay, now we got to cut out the first column. Okay, we got our genes, and now what? Yep. I think we can be confident that they're already sorted, but just for sanity's sake, let's sort it again, unique it, and count it. There we go. This gene called Y. I'm not sure what's going on there. Is there a gene called Y? Let's go to gene cards. I gotta know. It's like an existential question. Yeah, that's going to take a while to find out. I don't know. Okay, so there's another way to do this. Well, there's the way that Jim proposes one. There's also tools. I'm going to talk about a tool called group by and bed tools tomorrow that would basically short circuit this. Basically, at this point, we could just feed it into one command and it would have done all this. But anyway, I think it's just helpful to think about how you can. I think the reason I wanted to go through this, and I'm glad that someone asked a really hard question because this takes a while, is that I think it proves the point that with a few basic commands, if you just understand, the kind of mindset in Unix tools is they do one thing, and they do one thing well, and you can chain together these tools in different ways to answer pretty sophisticated questions with simple commands. Do you have a question?
So what if you're not interested in all genes, but like a certain type of gene, like calcium channels? How would you,
with the data set that outside of what we did here, just in general.
Well,
Okay.
or it's the same problem, but I'm only interested in, calcium channels or transmembrane.
So one thing you could do is if you could go to a website like Hugo or anything like that, just to identify genes that are in the pathway or have the function that you want, you could create a gene list, and then do you happen to know a gene off the top of your head that's on chromosome 22 that is involved? Probably not, but if you do.
I checked a couple things to see if there's anything.
Because one simple thing you could do, let's just do a control. You could, if you had a list, let's create a gene list. And I'm just going to put this one gene in there. But you can imagine that, let's make it a little more complicated and do a few genes.
TBX5.
Say again?
TBX5.
Okay. Is that on chromosome 22? TBX5? Anybody know any other ones? Why? Yes. All right. So now this list could be arbitrarily large. So what you could do is now you have this output. You can grep against a file. So basically it'll take this output and match. It'll only return lines that match any of the genes that are in your list. And the way you do that is you tell that this is a file of patterns that it should screen against. And so problem is that Y is present in some of these genes. So now what you got to do is you got to say -W. And that means I want to treat the pattern in here as a word, so it has to be separated by a white space. So it gets rid of these, but only returns that. Similarly, we could do something that I wonder if you can, I don't know if you can do that. No, you can't do that on Mac, never mind. But yeah, so if you had candidate genes that you're interested in, one thing you could do is just start at the very beginning when you grep here and pull out variants. You could just grep that against your list. So you're just starting with those at the very beginning.
How hard is it to generate a candidate list like that for an associated organ system or something like that?
I guess it depends upon the function, the type. How broad the search is, because essentially you could use things like gene ontologies to just search for a specific cellular function and pull out genes that fall you can easily. There's plenty of tools online that will pull out all genes that fall into that particular function, but it really depends upon how granular and how specific you want to be, or how broad the search is. And is there a sort of a key paper in that field that you could use to pull out, from supplemental figure 19, all the genes that they found in protein-protein interactions or something? Yeah.
I have in mind, I'm trying to figure out, based on what you just did, I'm thinking about something.
Yeah. Okay. Well, if you want to help, I'm happy to help out. So we have 10 minutes left until dinner, so we can either try and answer another question, or we can. I don't think dinner's open till 5:30, but or we can just stop now and reconvene after dinner. We're going to talk about how to use a piece of software that my lab develops called Gemini to solve some rare diseases with real data sets. So maybe let's just break now and we'll come back.
You said you had an easy way to do what we just did.
Oh yeah, okay. All right, so prove it. Right. So I think it was at this point that yeah. All right. I'll show you. Let me just do it first because I have to remember how to do it, and then I will explain what's going on. Oops, helps if you spell things right. Actually.
Can you save this terminal session as a text file and post it?
Mm-hmm.
See the process here?
Mm-hmm. Oh, what is it called? Do you remember, Jim?
Which one are you looking for?
I don't remember. Oh, yeah, here it is. Oh, never mind. Sorry. Huh? Oh, sorry. I'm breaking something. I'm not sure what. Hold on. Yes, fine.
Maybe you should check with the creator of
Yeah. You can tell how much real work I actually do anymore. It's kind of embarrassing, but I don't know. It's
Dash just by itself.
There's a dash by itself.
yeah, that's supposed to be there. I broke something. I'm not sure what. So basically, what would happen? I'll figure it out and I'll post the example later. But basically, what would happen is the group by command. Basically, you tell it what column or columns you want to group by. So essentially, it's saying, I'm asking it to group by the third column, assuming we've sorted by the third column. So that's why I was trying to sort by third column. And then it will, for this group, it will just count how many unique, you can ask it to count how many unique values there are in this column. So it's exactly what we did. It's just a shorthand way of going through all those changes. Yes, you can. This is sort of old convention, but this sorting, you define a key. So it's by default, and really old, I learned on really old Unix, and you had to be specific about the range of the values that were sorted. If you didn't do this in the old days, it would sort from 3 on. But here is basically sort from 3 to 3, so only on the third column. Yeah, I don't know. I'll try and figure out what I broke.
If you don't put anything after the sort, it just sorts on the first column. I think we did that a couple times.
It sorts on the first column, then breaks any ties with the second column, then breaks any ties with the third column.
Okay.
But if you tell it a specific column, it only sorts on that column, and it's so-called stable sorted for every other column afterwards, meaning that it just comes in the order that it was in the file if there were ties. Yep. Okay. Well, I'll try and figure out group by. Thanks.